firstly, I would like to thank Helen for inviting me to review the genetics of the intrahepatic cholangiocarcinoma in China. So, as we learned from the previous several talks, the intrahepatic cholangiocarcinoma, like other cancer, is a genetic disease. So the better understanding the genetic disease will help us to learn how the tumor grows and develop, and to develop effective therapies, as well as to develop methods for early detection. Before touching the base of the cholangiocarcinoma in China, I would like to span through the general cancer incidence in China. Next slide, please. Okay, see. So China is a big country. The National Central Cancer Registry data was properly standardized not until 2008. And this paper analyzed the data based on the availability in 2020, so including the 1,152 local registries, represented nearly 600 million of the national population. Unfortunately, most of the data condensed on the somewhere east part of China, so where the economic world developed a bit more richer. So still have a kind of bias in this case, but nevertheless, the top 5 new cases were diagnosed in 2020 in male mainly are lung, liver, stomach, colorectum, and the esophagus. In female are breast, lung, colorectum, thyroid, and stomach. However, the paper didn't mention anything about the instance of the cholangiocarcinoma. Even you don't need to think about the availability of the instance of the subtypes of the cholangiocarcinoma in China. So it's a big mind for us to dig out next. But the availability of based on the recent publications, it seems the intrahepatic cholangiocarcinoma is sustained or not much difference. But the global trends of the intrahepatic cholangiocarcinoma instance in China is increased. But I guess only based on the studies from a couple of the publications, most likely going to be from the east part of China or southeast part of China. However, the majority of the China might be not available. So now the risk factors for cholangiocarcinoma in China is quite similar to what the Professor Banalis reviews published in 2020. However, the chronic liver disease and cirrhosis because of the hepatitis B infection contributed to nearly 30% of the intrahepatic cholangiocarcinoma. And because so many complicated risk factors plus the so many genetic faults of the disease, it can be summarized by at least 14 hallmarks of the cancer. And this is just famous. I think in the field, everyone knows this reviewed. And these hallmarks are the therapeutic targeting. So here list of the sort of inhibitors have been developed. In the case of the cholangiocarcinoma, we know the two inhibitors specifically target to the isocitrullinate hydrogenase 1, so-called IDH1 mutants, and FGFR2 mutations. So in comparison about the molecular genetics of the intrahepatic cholangiocarcinoma in China, in comparison to recent publications in a Caucasian cohort, which is American data, and where the genetic alterations mainly including the IDH1, RIDA1A, TP53, and the FGFR2. A similar study in a cohort in Shanghai, again, very limited data, they found differently, where the most commonly identified genes are TP53, KRAS, and ARIDAI, whereas the IDH1 only represented 11% of the mutation, while the FGFR2 only 2%, suggesting the sort of ethnic group difference in terms of the genetic information. Now, the just recent publications, again from the same university, and invested heavily on this project based on the more omics studies, as mentioned by previous talks. So the collaboration between three national key laboratories spent nearly £500,000 on this project. So they used 262 paired clinical samples and analyzed the genetic profiles, of the genetics, transcriptomics, proteomics, and phosphoproteomics. They identified the common pathways and molecules, and they integrated this data and figured out the implications in the prognostic significance in these patient populations, and trying to propose the futures for the clinical target development. So I'm not going to take you to go through all the figures, but the highlights as such here. Gene signature, where the aflatoxin is the prominent feature in this population. We know the aflatoxin is a toxin produced by the family of fungi and in the foods like peanuts, and they are important in the factors contributing to the liver cancer. The second feature is they saw the frequency of the chromosome 14 long arm deletion, and identifying the several of the gene mutations, typically TP53, KRAS, IDH1, 2, and the BAP1. Then about the FGFR2 fusion is famous, and they found the downstream of the target of FGFR2 fusions, including the PTBN1 and the GRAB-B2. So all these signalings are important in the regulation of tumor survival. Interestingly, they found the fusions partners of FGFR2 is BICC1. What is BICC1? It's an RNA binding protein regulated embryonic development. And the breakpoint of the peptides between these two proteins appear to be very to behave a very strong immunogenicity. So implication for the vaccine development for the immunotherapy in the future. Furthermore, by stratification of the patients with the proteomic signatures and into the 4 groups. So S1 is a signature of the information, which is patient survival rate as the worst compared to other 3 subgroups, including the metabolisms or the cancer-associated fibroblast, as well as the biliary secretor. So finally, they identify the prognostic biomarkers, and most prominently they found, including the HKDC1, which is hexase kinase domain components 1, as the molecules regulate glucose metabolism, and in particular the glycolysis pathway. The other one is the SLC16A3, as the lactate transporter. So I will highlight a bit in the next few slides. So as mentioned by John this morning, so there are at least 40% of intrahepatic cholangiocarcinoma can be druggable, and this population is about that kind of the range of the rates. And this druggable genetic mutation, including the mutations, amplification, or fusions, and typically including the KRAS, IDH1, 2, FGFR2, and the MET. MET is another cancer driver here. So on the right, you can see the list of 16 gene mutations identified here, and KRAS appear to be the most frequent genetic alteration genes in this cohort. Now, they compared this data set with the other 2 data, like MSKCC, which is Memorial Sloan Kettering Cancer Center from New York. The other data set is ICGC, is an international consortium, cancer genomic consortium. And you can see the frequency of the mutation between these 3 data sets is very significantly different. And furthermore, the distribution of the mutational signatures and associated with driver mutations is on the list. And again, TP53 and KRAS are the top 2. Now, if they investigated the correlation of the mutation of the KRAS and P53 with overall survival rate after month of operation, they found the double mutation of the KRAS and the P53 patient survival rate is the worst compared to either wild type of them or one combined wild type or mutant. Yeah, so therefore they think you suggested they found the spontaneous mutation of both of these genes posed extremely poor prognosis with increased metastasis. Now, back to the FGFR2 have been mentioned frequently. And so as mentioned earlier, where you can see the fusions FGFR2 have so many partners, where the BICC1 has 11 cases here in this cohort. And I also mentioned the peptides between these two breakpoints have very strong immunogenicity, where it stimulates the normal T cells to secrete interferon gamma. So interferon gamma, we know it is important factors in the tumor cell killing secretory factors. Yeah. And they also highlight where the conserved peptides present here, and currently they are trying to use these two peptides to stimulate the tumor response to the immunotherapy. Now, finally, so as mentioned earlier, they found there are 19 proteins, and red expression is associated with good prognosis, whereas 15 proteins expression is correlated with worse prognosis. Intriguingly, where high expression of HKDC1 is correlated better patient survival rate. In contrast, high expression of the SLC16A1 patients get worse survival rate. So therefore, these two may be a good prognostic predictor for the clinical practice. Although the number of the patients are so small, but it could expand in the rest of the cohort in the future. So back to the clinical management, it's very similar to the ESMO proposed. So based on the 31 Chinese expert consensus on the surgical management of ICCA, and in 2020 editions, as republished in Chinese, I translate into English here. So just the same, either surgery or systemic therapy. So take-home message: molecular genetic alterations is very different between the ethnic groups. It's different between persons. Therefore, future personalized medicine is the way to treat the patients. Finally, I would like to thank AMMF fund our DPhil students. Thank you for your attention, being patient. Thank you.